Hello. This is the second part of our Chapter 17 lecture. So I'd like to begin at the same place that we left off, which is talking about reabsorption in the proximal tubule. And in the proximal tubule, we had been talking about how glucose can be picked up at the end of the proximal tubule and sent back to the capillaries. And I would like to show you the molecular mechanism for that. So here we see sodium, shown in orange, glucose, shown in purple, can be co-transported across the membrane, the apical membrane of cells that are in the proximal tubule of the nephron. So here we have the lumen, where with the filtrate coming out of Bowman's capsule can have glucose and sodium will be entering this cell following its concentration gradient. And remember, that has energy in it when a molecule is moving with its concentration gradient. That energy will be used to co-transport glucose against its concentration gradient. So now glucose is being carried in from the lumen into the cell. And the energy of the sodium concentration gradient is going to be carrying in glucose against this concentration gradient. And this concentration gradient for sodium is maintained by ATP-dependent sodium-potassium transport pump in the basal membrane of the cell. This will be pushing sodium against its concentration gradient to the outside of the cell, and it will be bringing in potassium against its concentration gradient. And what this is going to do is this sodium leaving is going to actually enter the capillaries in the kidney, and then the potassium ions are actually going to be leaving the capillaries and diffusing out and then being actively pumped into the cell. And then if we look at another carrier in the basal membrane, we can see there's a glucose carrier, and glucose will be traveling by a facilitated diffusion outside of the cell and then be picked up into the capillary. And this is the important mechanism of reabsorption that occurs in the proximal tubule, specifically looking at the reabsorption of sodium and glucose. And then the player that plays a role in this that's not part of the reabsorption directly is potassium. So potassium is leaving the capillary and entering the cell via the sodium-potassium pump that uses ATP hydrolysis to pump both of those ions against their concentration gradients. So the next part of the nephron that I'd like to move on to is the loop of Henle. So fluid is going to enter the loop of Henle, and we have it entering the descending limb, and then there's this big U-shaped structure, and then the filtrate will enter the ascending limb. And there's a lot of complicated things that are happening in these two different segments. So what I'd like to show you is happening in the descending limb is we have water leaving the filtrate. Water is leaving, but ions are not leaving. So what it's going to do is going to lead to a increased concentration of solutes as the filtrate descends down this loop of the loop of Henle. And we can see the osmolarity increasing from 300 to 700 to 1000 to 1200 on the bottom. And that is due to there being less water. For the same volume of solutes, they're going to be in a smaller volume of water, making the solutes more concentrated. So this critically happens because the walls of the descending loop of Henle are not permeable to salt. However, they are permeable to water. And the water is being drawn out of the filtrate via water channels called aquaporins. So aquaporins are in the membrane of these cells lining the descending loop of Henle, and the aquaporin allows water to be exiting into the interstitial space. And then in interstitial space, it can be quickly picked up by capillaries. And again, the exiting of water leads to more solutes being concentrated, having this very big increase in osmolality as the filtrate is moving down the loop of Henle. Then, what we see happening in the ascending loop of Henle is the opposite story. However, instead of seeing movement of water, what we're going to observe is the movement of salt. So in the ascending loop of Henle, we have salt exiting the ascending loop of Henle, and as salt exits, we see the osmolality is decreasing from 1200, go to 1000, 500, and decreasing as we go up the loop. So in the ascending loop of Henle, we have salt, NaCl, being actively pumped into the interstitial fluid in the ascending loop. And specifically what is happening is we see sodium moving down its electrochemical gradient. Okay, so here sodium is traveling with its concentration gradient, okay, being put into the, moving from the lumen of the filtrate that's in the lumen into the cell. And then, how is this happening? So just like we talked about before in the proximal tubule, the reason there's less sodium in the cell than in the filtrate is because there's a sodium-potassium pump that uses ATP to pump out sodium against its concentration gradient and to bring in potassium against its concentration gradient, right? So the ATP hydrolysis funds moving both of those molecules against its concentration gradient. This allows for sodium to be entering the cell with following its concentration gradient. And then this has energy, and the energy of following this sodium concentration gradient allows for potassium to be brought into the cell against its concentration gradient, and also for chloride ions to be brought into the cell as well against its concentration gradient. So before we saw glucose is being carried against its concentration gradient by the sodium moving with its concentration gradient. And now, in a different part of the nephron, now the ascending loop of Henle, we're now seeing that the potassium and chloride ions are being carried against their concentration gradient by the energy of sodium traveling with its concentration gradient. So now let's look at the other side of the cell. So I want to show you that since sodium is being pumped actively out of the cell, chloride ions are always going to want to follow the sodium. So they're attracted, and these will be basically passively leaving the cell, following its concentration gradient. And also potassium will also be leaving the cell, following its concentration gradient. So basically, sodium leaving the cell into the interstitial space will attract the chloride ions to follow into the interstitial space. And that's how we're seeing this net movement of NaCl exiting the ascending loop, entering the interstitial space. And notably, I want to point out that the ascending loop of Henle is not does not have aquaporins, and it does not allow water to exit the cells. It's only sodium and chloride that can exit the ascending loop. And then in descending loop, in contrast, there are aquaporins, and this only allows the exit of water. There is no mechanism for the salt to be leaving the cell in the descending loop of Henle.